Okay, now it's time to talk about the Krebs cycle. This is also known as the citric acid cycle. And this occurs inside the mitochondria, the matrix of the mitochondria. And up until now, we went through glycolysis and the preparatory reaction. And while glycolysis happens outside the mitochondria, the prep reaction is on its way in, and it's generating the fuel to turn this Krebs cycle or citric acid cycle. The thing that we're making is acetyl coenzyme A during the preparatory reaction, and this fuel will spin a series of reactions called the Krebs cycle once. Now, let's think about this. In glycolysis, we took a single molecule of glucose and we split it into two pyruvic acid molecules. Those two pyruvic acid molecules were converted into acetyl coenzyme A molecules. So each acetyl coenzyme A molecule spins the Krebs cycle once. That means for every molecule of glucose burned, it will spin the Krebs cycle twice. All right? And think about that. Remember, we went from one molecule of glucose to two molecules of pyruvic acid, which were converted into two molecules of acetyl coenzyme A, and then those went in to fuel the spinning of this series of reactions called the Krebs cycle. There are a couple other things we need to take into account here. We have NADH and FADH2. NADH and FADH2, we talked briefly about NADH in glycolysis. Now we're talking about it here again. It's NADH throughout these previous steps are all just coenzymes designed to carry this high energy electron attached to a hydrogen. Okay? FADH2 is basically the same thing. All right? There's really no difference except for it's constructed differently. And people often ask me, well, why did it show up? I don't know, a random assortment of molecules. And so the only place we see FADH2 is in the Krebs cycle. Now, here is the series of reactions, and you're not going to have to memorize all this, okay? So just relax. When you first look at this, just like when you first look at the glycolysis, you remember that it looked pretty complex, but we're just going to break it down into what's coming in and what's going out. Basically, what we're doing is we're going to take one acetyl coenzyme A molecule, and it's going to spin these series of reactions. As a result of spinning these reactions, we're going to get a few things. We're going to get some NADH molecules, and those are going to carry those hydrogens to the electron transport system. Then we're going to get some energy, ATP molecules, using substrate level phosphorylation in this phase. And we're going to get a bunch of CO2, carbon dioxide. This is where the majority of the carbon dioxide that we breathe out in our respiration is generated. Okay? And remember, these little lines represent a series of chemical reactions. So here, this is an NAD molecule that, in the process of these reactions, picked up the hydrogen. So it was reduced. In the process of all of these reactions, we generated CO2. Okay? FAD picked up some hydrogens. Those are also going to go down to the electron transport system. Now, let's watch it in motion. Okay, so there's food molecules, comes into the cell, goes through glycolysis. The pyruvate is generated in glycolysis, and it gets oxidized, and bingo, here's our prep reaction. Pyruvic acid to our acetyl coenzyme A, and we generate some carbon dioxide there, and we'll make an NADH molecule. Now, the series of these reactions, all right. Okay, so we're going to watch this go through, and I'll talk about it as we go. And hopefully it'll go. There it comes. Glycolysis produces pyruvic acid, which becomes, in the preparatory reaction, acetyl coenzyme A. And you can see the NAD picks up a hydrogen there. We get rid of some CO2. And he's talking. Now here is where we start going through these different reactions. And again, you don't have to memorize all of these different reactions. All you have to do is kind of know what goes in and what goes out. So there you can see some NADH was made from an NAD plus an H, and we kicked out a little carbon dioxide. Remember, that line is a series of chemical reactions. Bingo, we made some more. Here's where we made some ATP. That's substrate level phosphorylation. And here we make some FAD and some more NADH. So we take some FAD and add some hydrogens, and NAD and add some hydrogens. And that's one of the acetyl coenzyme A molecules. Now the second one will do that again. So we spin this series of reactions twice from that original molecule of glucose. Okay. So basically this is kind of what we put into it. We have some acetyl coenzyme A groups, and we're going to get some carbon dioxide out. We have some NADs and FADs, and we'll get some NADH and FADH, and we're going to make some ATP. I'm not going to ask you how many ATP are made in the citric acid cycle or the Krebs cycle. You don't need to know these numbers. Don't waste time learning numbers. What I want you to do is spend your time learning the material. Okay? Now we get to the electron transport chain. This is the most complex, and at the end we'll summarize all this stuff. So basically it's going to get all of these electrons from the NADHs and the FADHs. Basically what's going to happen, the NADHs and the FADHs are going to come and drop off these hydrogens with an electron on them, and then it's going to process those through a process called oxidative phosphorylation. And in that process, we're going to make the majority of the ATP we get from each glucose molecule. By far, we get far more ATP molecules from the ETS than we get from anything else. Okay? Here is basically kind of what it looks like. This is a series of reactions, and we're getting ATP. But I'm going to walk you kind of through this. Now here, we're inside the mitochondria. Let me take you back here. These little membranes is where the ETS is located. And what it is, it's a series of proteins embedded into these little things called crista. C-R-I-S-T-A-E. C-R-I-S-T-A-E. And so we're going to take a little picture right there. All right? Now here comes an NADH and an FADH2, okay? And they're going to basically detach the hydrogens. Now, when they go through these hydrogens split, what happens is a hydrogen, if you remember from chemistry, the chemistry chapter, is a proton orbited by an electron. So here at the very beginning, we're going to take this NADH and we're going to split each hydrogen atom into ions. The proton, which we're going to call a hydrogen ion, and then the electron. So they're split. This is positively charged. This is negatively charged. All right? Now, the electrons will be picked up by this guy here, and transported to the next protein. That's where we get the name electron transport system. Okay, so the electrons were split from the protons, and the energy from these electrons, a little of it was used to pump these hydrogens to the other side of this membrane. And here we used a little more energy. Okay, and pumped some more. And so this is going on. And so what these electrons are being used for is to pump hydrogen ions to this side of this membrane. This is a permeable membrane. So when you get a lot of hydrogen ions up here and not very many down here, they're going to try to diffuse across. Now, we talked about this in diffusion. All right? And they will diffuse across through this little guy. This is ATP synthase. All right. Now, right there, let's look at that. We've got a bunch of these hydrogen ions on this side split from these electrons as well. So each hydrogen brought by NAD or FADH2 is split from its electrons, so you'll get some hydrogen ions here and some hydrogen ions here. You'll have more up here because the excess hydrogen ions will use oxygen and split it to make water. That's called metabolic water. All right? And you get about 30% of your water needs just from the food you eat, just through breathing in and getting those hydrogens from glucose and generating water. So let's watch it again. We're splitting off hydrogens here. And we're making a concentration gradient. And they're flowing back through here. Now notice, as these go through here, they provide power. Okay? And there's an ATP. So it's sort of like if you've ever seen a water mill. All right? You spin the water mill, and as you spin it, you can use it to turn a grinding stone in a mill to ground wheat or corn into flour. All we're doing is we're going through here and powering this little ATP synthase, and that's an enzyme. Remember, anything that ends in ASE is an enzyme. Okay? And it's using this enzyme to attach an ADP molecule to another phosphate, ADP, so that's got two phosphates on it. Here's the third phosphate. And it goes up to this little ATP synthase molecule and makes an ATP molecule. And that's energy. And ultimately, most of these hydrogens will eventually be, just like we saw earlier, will be soaked up by oxygen to make water. Okay. Here's another version I want to kind of look at. Looks a little different. Here's the NADH. Splits into hydrogen and electron. There's an oxygen molecule. So when it drops off its hydrogen, which splits, it's an NAD. It goes back to the previous steps and just picks up more hydrogens. You can see the electrons being transferred along this series of coenzymes and pumping, using the energy to pump hydrogens across the membrane. And as these electrons come, they hook back up to their hydrogens to an oxygen, and we make water. ATP synthase. As they move through, we make ATP. Now, why do we want to split a hydrogen atom into a hydrogen ion and an electron and bleed the energy off slowly? Well, hydrogen has a lot of explosive power. Okay, it's flammable, it can explode, etc. But imagine a balloon, and you fill that balloon up as full as you can fill it. And if you just let all the energy out that's stored in the balloon in the form of that air, the balloon will just fly all over the room, totally uncontrolled in a chaotic flight. But if you slowly, let's say you put your finger on the opening to the balloon, and let's say there's some dust or eraser dust on a desk. You could slowly let that air bleed out and do work. You could blow all the stuff off the desk. All right? By controlling the energy output, controlling the energy that's there. And so this is just a way to harness the energy for cellular work. And here's a picture of that going on. See, NADH, series of reactions, drops its H. Okay. You don't need to watch. You can watch that proton pump if you want. You have this in your system. Now, here is kind of a better summary. So glucose makes 2 pyruvic acids. Before it moves into the mitochondria, see this shaded part is mitochondria. This is the cytoplasm of the cell. Pyruvate, as it moves across the membrane of the mitochondria, it is converted to acetyl coenzyme A. 1 molecule, 2 molecules, 2 molecules. Each molecule of this spins the citric acid or Krebs cycle once. And so it makes a little ATP. Okay? Now over here, throughout all of these steps, we get all these NADHs and FADHs, and they're moved over, and then eventually down here in the electron transport system, these hydrogens are split off, and the energy is used to make ATP. Now again, don't waste your time memorizing the number of ATP. All right, you can look at different places, you'll find different numbers for it. Okay, just remember that the majority of the ATP comes from the electron transport chain, and a little comes from glucose and the citric acid cycle. These come from substrate level phosphorylation. This comes from oxidative phosphorylation. And if you remember, we use oxygen in this stage. That's why it's called oxidative phosphorylation. Now, we are going to do a quick summary. Just one other point I want to bring up here, rather, of glycolysis. Remember, in glycolysis, you had to spend two ATP molecules to get this whole system started, to start breaking down the glycolysis. By the end of glycolysis, you produce 4 ATP, but you used 2 ATP to get the system started, so you only had a profit or a net of 2 ATP. Okay, and you get a few ATP here. Important points to remember: 4 ATP here, 2 ATP is your profit. That's your net. Also, remember, if you don't have oxygen up here, you go into, instead of producing pyruvic acid, you will produce lactic acid, and that's anaerobic respiration. That's how wine is made. That is the burning in your muscles when you are lifting weights and you're feeling the burn. You run out of oxygen. The majority of CO2 comes from the citric acid cycle. Both of these produce ATP through substrate level phosphorylation. This is the place you get FADH. This only gives you NADH. This only gives you NADH. The electron transport chain takes these NADH and FADH and converts them to the majority of the energy that you get. And here's the oxygen. being converted to water. Okay, at the beginning of the chain, you split the hydrogens into a hydrogen ion or a proton and an electron. And by the end of the transport chain, they're reunited as hydrogen atoms again and then attached to oxygens to make water. Let's talk about the metabolic pool catabolism. The easiest thing to generate ATP from is from carbohydrates. However, if you run out of carbohydrates, you can get energy from fat. You can break the fats down into carbohydrates. You go through a process that's called gluconeogenesis. Basically, the false making of glucose. And we store excess carbohydrates as fats. And we can break these fats down into carbohydrates. So if that's the whole basis of all the anti-low carb diets out there, right? If you don't eat any carbohydrates or very few, then your body will be, the first place it will go is to fats, and it'll start breaking your fats down. The problem with the carbohydrate diet is a lot of people don't understand that once you make a fat cell, you have a fat cell. So you can alter your diet for a while and you're eating just protein like meat, grilled chicken, and vegetables. You want to try to really cut down on the fat. And by grilling the chicken, a lot of the fat drops out if you cook it well done. And then these fat cells will empty. But if you go back to your old diet and start eating carbohydrates again, then bingo, they'll just fill right back up. Because here in this country, carbohydrates are the cheapest food and they're the most easily accessible foods to get. And that is a bit of a problem. Also fats. Think about French fries. French fries, literally all a French fry is it's a big slice of potato and you boil it in fat, and so all the water molecules are pulled out and all the fat molecules take their place. It's literally just a fat stick. That's why it tastes so good. Okay? And if you really start eliminating all your fat, the next thing you're looking at is proteins. You'll start actually digesting your muscles and your proteins to convert into carbohydrates. And you can do that. If you eat a lot of proteins, you do need to increase your water intake. By eating all proteins and vegetables, or even just all proteins, you can really produce a load on your kidneys. And so you need a lot of water flushing through your kidneys to wash that stuff out. All right, that wraps up Chapter 6.